honeybee nutrition, varroamites, and small hive beetles. I'm sure most of you, or all of you, when you register, you were able to see his full bio. I'm not going to read all of it. I'm just going to read part of it. That all of it is impressive, but I'm just the part that I'm most impressed with, and sort of also jealous, because I wish I was that gifted and talented when I was his age. So in 2021, he was recognized as one of the nation's leading top 30 youth scientific minds in the prestigious Broadcom Master STEM competition among 10 million students across the country each year. Amongst this pool of top 30, he was recognized as the first place national winner in the category of scientific research. In 2022, he was selected as the winner of the at-large BioGenius Challenge as one student from across the entire US, and qualified for the 2022 International BioGenius Challenge, where he was named the highest honors awardee. He is also a very active public speaker and actively uses STEM as a catalyst to make positive reform in the community. With that, Atreya, the floor is yours. Thank you very much for being here, and I'm looking forward to your presentation.
Thank you so much, sir.
You should be able to share the screen, Atreya. There you go.
Okay, so I hope you're all able to see this PowerPoint.
Yep, we got it. One more thing, Trey, before you start. As questions come in, do you want me to wait with the questions till the end, or do you want me to ask them as they come in?
It'd be better if we could keep them towards the end.
Okay, you got it.
Yeah, thank you.
Yep.
Okay, so thank you so much, Mr. Emarson, for the warm introduction. I'm happy to be with you all here tonight. So my name is Atreya Manaswi. I'm a 10th grade student from Orlando, Florida. I attend Orlando Science High School, and I'm going to be speaking to you all on small hive beetle biology and control. So I'm affiliated with the University of Florida's Honeybee Research and Extension Laboratory, and also the US Department of Agriculture. Now, before I get into my actual research and how I got into all of this, I'd like to begin with my backstory. So 4 years ago, I went on a short fishing trip with a friend and his grandfather, and my friend's grandfather is a beekeeper. So he began to tell us about the stories about his beekeeping practices and about how decades ago he would get dozens of barrels of honey. And he told us about how that season he had gotten nearly 3. So this was very shocking to me, and I went back home and I dug into the research and the statistics, and I did some brainstorming. And after that, I attended the South Florida Bee College, where I was inspired, and I stepped into the laboratory at the University of Florida, where I began my first research project. Now, in the top right, you can see an image of me at age 12 at the UFB lab. And so this was a laboratory study, which is a very comfy and a cozy environment. But stepping into the field in future years, I saw how hard it is really for beekeepers. There's a lot of sweat involved. It's very hard work. These hives become very heavy, and there's stings, obviously. So that really taught me to appreciate the value and the hardship that goes into beekeeping and how difficult it really is to put food onto our plates. Now, for the presentation tonight, it'll be broken down into 2 principal components. So the first of which will be on the biology of the hive beetle. This will be focusing on the background, morphological characteristics, and behavioral and social characteristics. And then the second aspect of this presentation will focus on treatment. So this will be twofold: chemical-based agents and then organic-based agents. And the research that I will be sharing with you that I conducted is on organic-based treatment for small hive beetles. Now, the takeaways I'd like you to have by the end of this presentation are firstly, what small hive beetles are. Secondly, what these small hive beetles do to your bees and inside your colonies. Then thirdly, what I learned from my research. And lastly, how you can apply my research findings into your hobbyist or your commercial beekeeping practices. So let's begin with the hive beetles biology. Now, Athena timida is the scientific name for these hive beetles, and they're actually endemic to sub-Saharan Africa. Now, this was prior to the 1990s. However, in 1996, the first report came from a port city in South Carolina, and this was a small hive beetle reporting. Now, in 1998, the first official infestation came from Florida, which is my home state. Now, one thing to note is that these small hive beetles are a very little threat to the African honeybees, which is where they are endemic. However, the European honeybee towards the European honeybee, these small hive beetles are a major threat. The European honeybees cannot defend against these hive beetles. And because of this and beekeeping trading through different materials, as you know, in the US, we have a very large migratory beekeeping operation with nearly 2/3 of our hives going to California each year for pollination of almonds. So this is essentially a frenzy for different pests and parasites to spread between these hives. And as a result of this, these hive beetles are simply spread all across the globe. Now, these beetles are described as opportunistic scavengers in the scientific literature. And what they actually do inside the hive is they destroy honey and pollen stores and they damage cappings and comb, which significantly stresses the colony. So this is an image of an adult small hive beetle. And on the right, one of these is a male and one of these is a female. So I won't tell you which one is which as of yet, but something interesting to note is that you can actually sex these beetles. And I've done this a lot in my research. What you can do is, as you can see in the bottom right, is take the body of the small hive beetle and then squeeze, and one of three things will happen. So the first thing that can happen is that a very long appendage will shoot out. And you may be inclined to think that this is a phallus and that's a male, but you'd be wrong. And so this long thing, this appendage that shoots out is actually called an ovipositor. So this ovipositor is basically an egg-laying machine gun, and it indicates that it's a female. So the female beetles can take this ovipositor and stick it into cracks and crevices within the hive to deposit their eggs. The second thing that can happen is that a very short appendage will shoot out or nothing will shoot out at all. So this would indicate that it's a male. And the third thing that can happen is you squeeze too hard and the beetle simply explodes. So that's how you can sex the beetles. And so I did this very often in my research, and it was always a fun thing for me to do. Now, these are some pictures of other types of beetles that can show up in your colony. So here on the left, you have a sap beetle and a pollen beetle. Now, this sap beetle and this pollen beetle both are similar at first to the small hive beetle. However, if you look closely, these beetles have different colors on them and markings, and they're also different in their size. For example, this sap beetle on the bottom is 2 to 3 millimeters in length. So compared to the small hive beetle, which is 4 to 6 millimeters in length. Now, I say this to say that just because you have a beetle that shows up in your hive or an infestation, it is not a call for concern, and it doesn't necessarily mean that you have small hive beetles. There are other types of beetles that do show up in colonies. The best thing to do is to compare these beetles side by side or to call your local bee inspector and ask them about this. Now, this image on the right here is of a large hive beetle. So this specifically, this species is one that is endemic to Africa. Now, these large hive beetles don't have a particular species, but rather each region has their own assortment of these large hive beetles. And they get their name because they are large in comparison to these small hive beetles. And the small hive beetle was actually named small hive beetle because it's smaller in comparison to this. So this large hive beetle is 20 to 23 millimeters in length. And as I mentioned, small hive beetles are 4 to 6 millimeters in length. Now, I'd like to discuss the life cycle of the small hive beetle, and there's 4 primary stages in the life cycle. The first stage is the egg stage, and so these eggs take 2 to 4 days to hatch, and once they hatch, they emerge as these larvae. So these larvae are really the root cause of the damage that small hive beetles cause inside the hives. These larvae consume the honey, the pollen, and they tunnel through the capping and the comb, and they consume it. And they do this for 7 to 10 days once they essentially fatten themselves up. Now, once they're done consuming these hive products, they exit the hive and pupate in the vicinity within the soil. So this is an example of a pupae that's been dug up. These pupae pupate in the soil from anywhere from 3 to 6 weeks. And then after that time, they emerge as adult small hive beetles. So it takes 24 to 48 hours for these beetles to turn into this reddish brown hue. Now, one important thing to note here is in optimal conditions. So for the eggs, it's 2 days. For these larvae, it's 7 days. And for the pupae, it's about 3 weeks. This 2 plus 7 is about a week, plus this third week is 4 weeks, give or take. So it takes 4 weeks for these eggs to become these fully matured adults. Now, that's important because 4 times 6 is 24, and the life cycle of this hive beetle is 24 weeks. So this shows that in optimal conditions, you can see up to 6 consecutive generations of these beetles within a hive at any given point. And in addition to that, a single female can lay 1,000 to 2,000 eggs upwards in a single lifetime, and that's a single female. So this really goes to show that if these small hive beetle infestations are not controlled, they can really spiral out of hand. And so it's very important to mitigate the effects of this pest. Now, as I mentioned, the larvae are the root cause for most of the damage that small hive beetles cause. These larvae appear very suddenly and in large numbers, often in the hundreds or the thousands. So this image in the middle here is a larval infestation. And when this occurs to this extent, it's actually called a slime out. So when a slime out occurs, the brood rearing will cease entirely and the population will dwindle. In fact, something that can happen is the bees will abscond entirely from the hive, so they'll just desert the hive. And so these small hive beetles are often considered a marker for CCD, which you may be familiar with, or colony collapse disorder. So there's this yeast that is associated with the larvae of the small hive beetle. And in the bottom left, you can see this yeast under the microscope. So this yeast is called Codima omeri or K. omeri, and it's pretty well documented in the literature. But essentially what this yeast does is it causes fermentation of the pollen and honey that exists within the hive. Now, when this yeast begins decomposing pollen and honey, it produces this volatile odor compound called isoamyl acetate. And this is actually a honeybee alarm pheromone. So when isoamyl acetate is released by this yeast, it serves as essentially a calling signal to other small hive beetles in the vicinity of that hive and tells them that there's a surplus of food in this region. Now, there's really two telltale signs of a sufficiently bad larval infestation or a slime out like the one you see in this middle image. So the first of which is that when you pop open the super on your hive, you will smell a rotting citrusy odor. And the second will be that you will see honey bleeding out from the front of the colony, as you can see in this image on the right. Now, an interesting behavioral characteristic I would like to note, and this is an interaction between the hive beetle and the honeybee, is something called a jailing phenomena. Now, this isn't an official term, but this is also well studied and well documented. But essentially what happens is that these honeybees begin to corner small hive beetles into cracks and crevices within the hive, and they essentially jail them as if they've been in cells. So you have these guard bees that are making sure that these beetles stay in that region and do not roam around the hive. So the interesting thing here is that something that occurs only 10% of the time is that this beetle is able to trick the honeybee into thinking that the beetle is actually a honeybee. So you can see this in the bottom right. What the beetle will do is it will take its antennae and actually antenate against the mandibles of the honeybee. So this causes the honeybee to think that the beetle is actually a honeybee, and so it will start regurgitating food and feeding it. Now, as I mentioned earlier, this is one of the characteristics that makes this beetle a very, very smart predator of the honeybee. Another thing I'd like to discuss is the distinctions between wax moth larvae and also small hive beetle larvae, because at first glance, without knowing, they do look very, very similar. But some easy ways to identify which one is which is for wax moth larvae. Firstly, these wax moth larvae are covered in long hairs, as you can see here, all over their body. They also have 8 prolegs here in the middle and 6 legs near the head. And the biggest characteristic is that these wax moths create cocoons and webbing within the hive. For small hive beetle larvae, on the contrary, these small hive beetle larvae are much smaller. They're merely a quarter of the length and a quarter of the, or half of the length and a quarter of the width. So they're much, much smaller. They also only have 6 legs in the front of their head, and their abdominal segmentation is much more pronounced. So some good general apiary practices to manage small hive beetle infestations are as follows. Firstly, when exchanging or combining comb between different hives, it's very important to be careful because you can actually introduce small hive beetle eggs from outside the colony into that colony. Also, when removing old frames or hives, or even adding new frames, it's important to not add rotten, cracked, or damaged comb. So this dilapidated comb or frame makes for very good hiding spots for these beetles. As I mentioned earlier, these female beetles are able to take their ovipositors and deposit eggs into these cracks and crevices, which is suitable for their reproduction. Now, when pulling honey from the colonies, it's very important to extract as quickly as possible, preferably within the 2-day period. These beetles will actually, the beetles within the frames will consume the wax cappings and then start defecating inside the hive. So this ultimately causes fermentation through that yeast. One thing that some beekeepers do is they freeze their frames. And so this is another thing that you can do to mitigate the beetles defecating inside of the frames. An easy thing that you can do is exposing the hives to more sunlight. So the beetles are repelled by sunlight, and the bees, on the other hand, are aggravated, but they don't mind as much. So this is an easy way to help manage these infestations. And lastly, when creating nucleus colonies, or even when adding new frames or new supers onto pre-existing colonies, it's important to only add as much to the colony as needed. So only adding as much as you see the growth of that colony being, because in this process of adding more frames or more supers than the colony actually needs, you're giving these beetles unpatrolled room essentially to wander throughout the colony. So if the colony is weak, especially, these bees will not be able to monitor where the beetles are or what they're doing. And so this will give them a better chance to reproduce. Now I would like to transition into the second aspect of this presentation, which focuses on treatments. I'll be beginning with these chemical agents. So there's two main agents for small hive beetle treatment in the industry. The first of which is checkmate strip. So this has a 10% active Comophos ingredient. And the second is Guard Star. So this has a 40% active permethrin concentration. Now for when these different chemical agents are actually applied, Comophos is applied as a strip. And so Comophos is also used, or checkmate, is also used for Varroa treatment. But for small hive beetles, it's applied as a strip, normally attached to cardboard and placed on top of the frames horizontally. And this is applied during stages 1 and 2 of the life cycle you see here when the beetle is an adult. So when these beetles come in contact with that strip, or even the honeybees come in contact with that strip, the chemicals disperse throughout the hive. And so eventually it kills these beetles. And the permethrin, which I mentioned, Guard Star, is applied during stage 5 of the pupation in the soil. So it's applied as a soil drench, and whenever these pupae come in contact with it, they simply die. And so these chemical agents have become of concern recently because, especially with Comophos, which is used inside the hive, they can leave residues inside hive products such as propolis, wax, royal jelly, comb, and honey. Now, transitioning to these non-chemical or organic and biological treatment agents, there are a few that do exist. Now, there are attractants that exist, and so these can be placed inside in-hive traps. So this trap, what you see here, is a beetle blaster trap. This is placed in between the frames of a colony, and it's filled about halfway with this attractant or this solution. So apple cider vinegar, sometimes in combination with mineral oil, is something popular that beekeepers like to use to treat for these small hive beetles. And so how this mechanism actually works is that this trap is placed inside the hives to essentially bait the beetles. And so the beetles are lured in by the aroma provided by this attractant. This image here, which has a blue background, is a baseboard trap. So again, treatment solution can be placed in this and the beetles fall. This is diatomaceous earth, this third image. And so this is sprinkled around the vicinity of the hive. And whenever these small hive beetles come in contact with this, it dries out the cuticle. And this eventually kills them. This last image here is of a nematode. So these types of nematodes are classified as entomopathogenic nematodes. So these nematodes essentially are released inside the soil, and they make their way to the pupae of the hive beetles, or the pupae hive beetles, and they make their way specifically into the gut eventually through an orifice such as the mouth or the anus. And inside the gut, they function as a parasite, so they eventually kill the beetle from the inside. Now, I'd like to discuss the advantages, the disadvantages of the chemical agents, and then also the proposed advantages of the organic treatment solution that I researched. Now, some advantages of these pre-existing chemical agents are that firstly, they're highly effective, and then secondly, they show very fast results. However, they do pose a series of disadvantages. And so the first of which is that they're extremely unaffordable, especially on a large commercial scale. Secondly, they do pose a risk of soil, water, and environmental contamination. And lastly, they have major adverse health risks to not only the honeybees themselves, but also wildlife, aquatic life, and humans. Now, the proposed organic treatment solution that I was researching was shown to be affordable and sustainable, highly efficacious for small hive beetle capture, naturally biodegradable, and environmentally friendly, and it poses no risk whatsoever to wildlife, aquatic life, humans, or the honeybees themselves. So what was my research? My research can be broken down into two main phases that I'll be sharing. So the first phase or first year of my research investigated 7 organic and inexpensive small hive beetle treatments. And so the second phase or second year of this research developed a novel treatment based on beer. And so beer was discovered surprisingly to be the best agent by far from year 1 of research. And so year 2 essentially focused on creating a refinement of beer. And so the chemical breakdown of beer was analyzed, and specific chemical compounds were chosen from beer and then concentrated in the form of a blend. And so the goal of the second year of research was
Atreya,
to create...
I have to ask this. I'm sorry to interrupt, but I have to. What kind of beer?
This is a lager beer, Miller High Life.
Okay, thank you.
Yeah. So this blend, the goals for this blend were to make something that would be even more affordable, and then secondly, even more effective than beer itself. So let's dive into it. So for phase 1, this research analyzed 7 organic agents. And so these were apple cider vinegar, which was the control of the study, mango puree with boric acid, cantaloupe puree with boric acid, and both of these had a boric acid concentration of 2%. And baker's yeast, peanut oil, grapeseed oil, and beer. So these specific agents, you may be wondering, how in the world were they chosen? They were chosen based on literature findings. So it's been shown that the small hive beetle can feed and reproduce on tree sap, rotting fruits, and other such odorous compounds. And so based on these hypotheses, these odorous, organic, and inexpensive agents were chosen for the research. Now, they were placed inside these mini beetle blaster traps that I mentioned about halfway. And so this image on the bottom right you see here is of cantaloupe puree. All these black specks are beetles that have been captured and killed inside the trap. Now, these are some images from year 1 of my work. And by far, my favorite memory is probably me counting beetles in my mother's kitchen. So about that story, this was during the pandemic in 2020. So I didn't have access to any commercial laboratories to get into for my beetle tabulations and countings. So I had to create a sort of mini lab in my mother's kitchen. And I remember one time I was cutting these traps open and some beetles were alive and they got loose. So my brother and I ran around with fishnets in hand trying to capture these beetles. And so they can fly for miles on end. And it was a very difficult process. So till this day, we still might have beetles in my household. We'll never know. Now, this image on the left here is of me in a beekeeping suit. And you may be thinking, I have my protective equipment on, I have my beekeeping suit, my gloves, I had a smoker as well. And you're probably thinking, what a responsible young beekeeper, but that's completely incorrect. I was stung dozens of times within the field, sometimes on my legs, my arms, even on my neck. So this is a very, very painful experience for me and also very eye-opening. But I enjoyed it. I enjoyed my field experience very much. Now, this image on the bottom right here is of beetles being captured inside this beetle blaster trap. And the solution you see here is beer. So again, all of these black specks are beetles that have been captured. And so how this study was executed is that there were 24 honeybee colonies at an apiary site. And this study was run for a 6-week period during fall. So each week I would take these traps and place them inside the hives in the upper super, and then the second super. So an upper super and then the second super, and leave them in for a week. And then the week later, or the week after rather, I would come in and extract these treatments and then replace them according to a generated randomized schedule. So this was a randomized controlled trial. So that's how the tabulation was done. And when these traps are captured each week, a weekly count was established for each of the replicates and the treatments. And so that was done for a 6-week period. Now, after all of that data collection, extensive statistical analyses were run to corroborate the findings. And so I won't dive too much into this, but essentially what's important to note here is that this is a test that basically compares the control and the other agent, the study that you want to look at. So in this case, apple cider was the control of the study. And so apple cider was being compared against mango mix and cantaloupe and yeast and peanut and etc. So this control is being compared against the other treatments, basically seeing whether this control or the other agent was significantly different from one another. So apple cider, so in the comparison for apple cider and mango mix, you can see this orange arrow. And so this is marked as significantly different. And it means that either the apple cider or the mango mix was doing much better. And it's assumed that it was the mango mix, because apple cider was the control. Now, apple cider here is being compared against cantaloupe and then yeast and then beer. So all these columns that have an orange arrow above them were shown to be significantly different. And this basically means that mango mix, cantaloupe, yeast, and beer were more effective than apple cider throughout the course of this study. And peanut oil, grapeseed oil, and the control, which was just a blank trap, were not shown to be more effective than apple cider. Now, this still didn't quantify which of these specific agents was the best by far. This test just showed whether a specific agent was better or not than apple cider. So to actually find out which one agent was the best out of all of these, a series of tests had to be run. So the first of these was an ANOVA. So this is an analysis of variance. And then this was followed up with a Tukey HSD test. And basically, what this test found was that of all of the agents, and what this test does is it compares all of the agents against one another individually. And this is a very rigorous analysis, but what it showed was that the beer, which was treatment 7, was significantly different by a very large margin. Now, visualizing this and moving away from the world of statistics and numbers, you can see that beer is in deep blue here, and cantaloupe mix is in gray. So cantaloupe mix was the second most effective treatment. You can also note apple cider, which has a beetle capture of 11 compared to beer's 198 beetles captured. So what's important to note about this Pareto chart is not just these bars, which show the individual capture, but something called the 80-20 rule. So this 80-20 rule basically states that if you start at this 80% mark and then draw a line horizontally and trace down, that there will be 2 segments here, this one on the left and this one on the right. But what this rule says is that if you look at the data on the left here, 20% of this data on the left accounts for 80% of everything that's been captured. So I'd like to reiterate that. 20% of everything here on the left accounts for 80% of all the beetles captured throughout the course of the study. So this really goes to show the significance of the beer treatment and also to some extent the cantaloupe mix compared to the rest of the agents. This is the box and whisker plot for the trial. You can see beer here again is in deep blue, and the X represents the mean. So I'd like you to pay attention to this, please. Looking at the mean, this shows a higher consistency in the beetle capture throughout the course of the 6 weeks. So the X for the cantaloupe mix, which is the second best, is also shown here. And you can see the X for the beer is much higher than any of the other treatments. Now, ultimately, the conclusions from phase 1 were that beer was 33 times more effective in its beetle capture than apple cider vinegar, and it was the most effective treatment by a large margin. Moreover, beer is shown to be very cost effective and costs merely 5 cents per hive on average. Beer is also widely and readily available, and it does not harm the bees and only harms the beetles. So I assure you, using this beer treatment inside your hives, you won't end up with any drunk bees. Now, if you didn't believe me on that cost comparison, I'd like to highlight something here. So not only is beer 33 times more effective than apple cider, but it's also 3 times as cheap. This is just an image from Walmart.com. You can see Miller High Life, which is the lager beer that I used, is merely 7.3 cents per fluid ounce, compared to this apple cider vinegar, which is 19.8 cents per fluid ounce. So you're seeing that this beer brand is about a third of the cost of apple cider. So it's more effective and it's also more affordable. Now, moving on to the second year of this research. The second year, or phase 2, focused on the development of a novel volatile blend based on beer's chemical composition. And so a volatile is essentially just any compound that evaporates very readily at room temperature. And so the goal was to create a refinement of beer in the form of a more concentrated solution using specific volatile compounds. And so this blend, the goal was to make it cheaper and more effective than beer. Now, how did I actually go about doing this? So the first step in this process was actually extracting these chemicals from the beer. And so what you see here on the left is something called headspace sampling. And so this is where the volatiles are extracted. So this is a beer bottle, and this white tube that you see here is called an adsorbent. So this adsorbent has been attached to a tube that's connected to a vacuum. So when this vacuum is turned on, the gaseous compounds that are inside this beer bottle are actually taken, sucked, and then trapped inside this white tube or adsorbent that you see here. And so that adsorbent then stores those chemical compounds inside of it. Now, after these chemicals from the beer have been stored inside that absorbent, they're then placed in a GCMS, or gas chromatography mass spectrum, which is a mouthful. But essentially, when that adsorbent is placed inside this GCMS, it's heated to a very high temperature of 220 degrees Celsius. And this causes the release of the chemicals from that adsorbent into a column inside this machine. So this is a very long circular tube that's 10 meters to 100 meters in length. So it's very, very long. And so what happens is, as these compounds are released into that very long tube, they begin to separate based on their weight. So they're essentially, the heavier compounds begin to sink and fall back, and the lighter compounds begin to move faster through that tube. So at the end of that tube, there's a detector. And so these lighter compounds reach that detector first, and they're picked up first. So this is important because this GCMS produces a reading here, which you see on the right. And this reading is called a chromatogram. So this chromatogram on the x-axis you see is minutes. So this is time. And so the closer you get to the left, the quicker these compounds have been picked up, and likely the lower their molecular weight is. So the lighter they are. And so I mention all of this about molecular weight and time because the compounds that have a lower molecular weight are carried more easily in the air, and thus they're picked up more easily and readily by the beetle compared to these heavier compounds that tend to sink in the air because they weigh more. And so it was these compounds with a lighter molecular weight that were a focus for a blend because they could be easily picked up by the beetle. And so through these chromatogram readings, 4 chemical compounds were chosen ultimately. They were shown to be very light in their weight and also abundant in all of these samples. So there was a very high presence of them. Now, after this process, after those volatiles were identified, they were tested in something called an EAG, or electroantenography. And so what's happening here is basically there's 3 focal components. The first thing here is that there's an electrode, and this electrode has the small hive beetle antenna on it. So these antennae were excised under the microscope. And as I mentioned, these beetles are very, very small, 4 millimeters to 6 millimeters in length. So doing this under the microscope was like neurosurgery for me, but on small hive beetles. So it was a very fun, but also somewhat tedious process. And I did get the hang of it after a few tries. Now, after those antennae have been extracted, they're live from anywhere for 3 to 5 minutes. So time is of the essence, and they're placed onto that electrode with some gel. Now, this tube here you see is a glass pipette. And so this tube has some rolled filter paper inside of it that's been dipped inside the compound that's being tested. And then the last component of this is this other tube here, which is connected here. So whenever this tube is connected to a foot pedal, and so that whenever that foot pedal is pressed, air is pushed through this tube, and then it flows through this tube and carries the volatile compound on this filter paper and exposes it to the antenna. So what's happening here is a specific compound can be taken, and then the response of the beetle antenna can be observed on that compound. So you can basically see how the beetle was responding to individual compounds. And so when that beetle detects that response, the electrical signal is amplified through this detector to a computer where a reading is generated, and that reading can then be analyzed. And so you can see if the beetle is giving a negative or a positive response to a compound that's being tested. So it's a very sophisticated process and something very useful that can be implemented. And it was very practical in this case for the synthesis of a blend. So after analyzing those results, a blend was formulated and an extensive field trial was run. So this field trial consisted of 28 honeybee colonies, and it was run over an 8-week period at 3 different apiary sites. So here you can see this image on the right. And so this is of the oil blend that was synthesized. And so this is shown to be the most effective agent by far. And so all of these black specks you can see here are small hive beetles that have been captured. Now, these are the statistical analyses on the data, and I don't want to dive too much into this, but basically there were 4 treatments that were tested: beer, an oil water blend, which had a water as the base, then the oil blend, which had oil as the base, and then the control, which was just plain old vegetable oil. So after extensive statistical analyses, the oil blend was shown to be the most effective agent by far. And so we can look at this visually here again with the Pareto chart. You can see the oil blend in gray, and it's much better than beer and the oil water blend, as we can see here. Again, applying the 80-20 rule, we can look at this 80% mark, trace here horizontally and then down vertically, and we can see the significance of the oil blend compared to the other treatments. Now, ultimately, the conclusions from phase 2 were that the oil blend treatment was very effective by far margin, and it was shown to be 5 times more effective than beer and merely half the cost. So why should you use my findings, and how can you actually implement them? So unfortunately, at this stage, the oil blend can't be synthesized, so you do need laboratory equipment to synthesize that. It's not available commercially as of yet, but what you can use is beer. So beer does not harm your bees in any way. As I mentioned, you will not end up with any drunk bees. And beer can also be used in any in-hive trap that requires some sort of trapping solution as an attractant. So beer is also very cost effective, as you can see in this comparison here, and it was shown to be much more effective than apple cider as well. Now, the key takeaways for small hive beetle management are really, firstly, to have good apiary practices. So maintaining cleanliness within the hive, exposing your hive to sunlight, not ensuring that you're not placing too many frames inside the hives or too many supers to allow for your bees to patrol properly inside the hive. And then using treatment, whether it be ultimately up to you, chemical or organic-based treatment methods to treat for these small hive beetle infestations. So it's ultimately the synergy of these two, good apiary practices and treatment for these small hive beetles, that reduces beetle populations to a low and a manageable level. These are some resources that I can definitely share with Mr. Marson after the presentation if you'd like to take a look. So these first two, this is a PDF and then this is a website. So these both have good information on small hive beetle biology and then also some control mechanisms. This last link is to the UF Honeybee Lab page, which contains some great videos, not only on small hive beetle management, but also on other tips and tricks for beekeeping practices. So that's all I have for you all tonight. Thank you so much for your time and attention. Before I open up to a Q&A, I do have one small request. You can do this now or afterwards, but if you don't mind opening up your phones to the camera app and holding it up to this QR code, this will take you to a very short survey. It has 2 questions and it's just about how you thought I did with my speaking and my PowerPoint. So I greatly appreciate it if you could fill this out. It's very useful to me for future presentations. So that's all I have for you all. Thank you so much. And I'm open to Q&A now.
Thank you, Atreya. And if you want to leave that slide up, feel free for the time being. So there's a few questions that well, more than a few, but quite a lot of them sort of overlap with each other. So the most common question that was asked was, so obviously you did this in Florida, right?
Yes.
Yeah. So the most common was what time of the year and how long was the total experiment trial?
Right. So the first year of this research was conducted for a 6-week period, and it was conducted in the fall using 24 honeybee colonies. The second year of research was conducted again in the fall with 28 honeybee colonies, and this is at 3 different apiary sites. So this is a few different sites across Florida. Now, fall is a very good time to test for these beetles because this is really where they're at their peak.
That makes sense. All right. Let's see. Where are you getting your small hive beetle in hive traps? That looks like something that could be easily 3D printed.
Right. So these are actually available commercially. And so Beetle Blaster is sold. That's actually the name of the trap. I have actually looked into 3D printing something like this. And so it's been something that's in the back of my mind, trying to design a 3D trap, because that would ultimately lower the cost of these in-hive traps significantly. So that's something I do want to investigate in the future.
Somebody asked if you could use multiple of those different attractants in a hive, beer and the oil-based one, or would your recommendation be to stick to one?
Mm-hmm. Yes. So I would definitely stick to one. What the problem you'd run into with keeping multiple attractants within the hive is that, especially in the heat, these compounds would begin to conflict and their aromas would mix up with one another. So sticking to one type of treatment, whether it be the beer or the oil, if you'd like, keeping that and having more of those traps inside the hive would be more beneficial. Maybe having one in the upper super and then one in the second super.
Okay.
And so, yeah, the reason I mentioned, and I'll explain why they're placed in the upper supers, is basically because the beetles tend to move away from the brood chambers, which is where you have a lot of bees, and towards the top side of the hive, because this is where less of the bees are generally. So it's easier for them to go unpatrolled. And so that's why placing your traps in the top supers of your colony will yield the highest beetle capture, because this is where the most beetles actually sit and roam.
And I know you mentioned that this was done in the fall. Somebody's asking about what's the population of the hive. So I'm assuming that since you're done in the fall, the population is not, the bee population is not at the peak, but the small hive beetle populations are at their peak or just reached their peak. Is that correct?
Yes, so the colonies I actually experimented with for year 1 of my research were pretty strong. So the beetle populations were not soaring, but they were at medium level. However, the hives were quite strong at that time. And the second year of research, I experimented with 3 different apiary sites. So there was one site which was very weak, and we were seeing a very high beetle capture in that location. But even across the other 2 sites, which had relatively stronger colonies, there was still consistent beetle capture. So that does show that this agent is effective.
Sort of a follow-up question, and not directly related to your research, but maybe you've seen by your observation, maybe you know the answer. Somebody said that it always seems like there's a hive or 2 in the apiary that seems to attract the most beetles compared to the other ones. So are beetles attracted to some hives more than others, or is it just random luck?
Right. So that's a very good question. And so these beetles actually emerge from within the colonies. So once they pupate outside of the soil, they'll generally go inside a colony and stay there. So it's generally inside that colony itself that you'll see the capture coming from. If an odor is generally not strong, so strong that it can attract beetles from very distant locations from outside that hive into that hive itself. And so basically, what you're seeing here is weak hives, which have a very high beetle population. And so I saw this as well in my research, is if you have a very weak hive and you place an in-hive trap inside of it, you're bound to see a greater population of beetles inside that trap, dead beetles, because that hive itself has a much larger population of these small hive beetles. And that generally indicates that it's weaker, because these small hive beetles are generally considered a secondary pest. So they pose a major threat to these already weakened colonies. And these stronger colonies are generally able to defend more well against them as the bees are more active and able to patrol. So if you have a high beetle population, it generally means that you have a relatively weaker colony. That's not always true, but most of the time it's true.
How widespread or how common is it to have noticeable population of small hive beetles in hives in Florida at any given time?
In Florida, it's a very, very common pest, and especially in the South. So something that you notice with these small hive beetles, and as I mentioned earlier, is when they pupate in the soil, it's ultimately the moisture of the soil that helps them with their reproduction. So it's Florida, we have very, very moist soil, and we also have a very high humidity. So these are perfect conditions for beetle reproduction. And given that I'm living in this state and all the experiments were conducted in this state, this is actually probably one of the locations where you would have a very, very high population of beetles at any given point in time, because the conditions in the environment are very suitable for small hive beetle reproduction.
Gotcha. All right. Can, can flat beer be used, or does it have to be fresh?
Sorry, what was the question?
Can flat beer be used, or does the beer have to be fresh?
So I didn't experiment with flat beer, but I popped open a new can every time I was working just to be consistent throughout the course of my experimentation. So that isn't something that I've experimented with, but I do know that the odor can increase. The carbonation will obviously go down, but the odor may increase over time. So that's definitely something you could experiment with and see how that does if you'd like to do that.
How effective is handpicking and destroying adult beetles during hive inspections as opposed to trapping them?
Right. So ultimately, as I mentioned, if you have one beetle, even one female beetle, she can constantly be laying eggs. And there's only so many beetles that you'll be able to see when you open that super. And generally it's on the lid. You might find beetles crawling around. So handpicking them and crushing them is useful because you're obviously getting rid of those beetles. But the best thing to do is implement an in-hive trap. So you have surveillance, and so that these beetles can be lured in and killed throughout the clock, like around the clock. Basically, implementing an inexpensive agent such as beer or apple cider and using an in-hive trap is something very easy that you can do.
I sort of a side add-on. Actually, feel free to pitch in if you know this, but I've heard or read somewhere that small hive beetles can be vectors for viruses. They can carry viral spores that are associated with varroa mites, but they don't directly transmit them. So killing them, squishing them on the inner cover and then leaving them for the bees to clean up and remove can potentially lead to increase in viral loads within the colony. Have you heard that?
Yes, I have heard about that. So the research in that field is not very extensive. So there are a few papers that do suggest they may act as vectors. Now, it isn't direct transmission, obviously, but squeezing these beetles and then placing them back inside the colony would definitely lead to that. So if you wanted to squeeze these beetles and kill them, you could have a container and then keep them inside that container and dispose of them. But placing them back inside the colony, that would not be ideal.
Gotcha.
Yeah.
Does the comaphorol, so COMAPHORL, kill any honeybees? Comaphorol?
Comaphos, yes.
Yeah.
Comaphos, Cumaphos, yes. So
Okay.
yeah, so the comaphos, which is the 10%, and it's applied as a strip inside the hive, there is research on that. And so it doesn't directly kill the bees. It's not toxic enough. It does kill the insect itself. But over the long run, it has shown to have some slight effects on the honeybee. And so these include things like limiting foraging activity, decreased learning behavior, memory. So these are all long-term effects that have been shown in correlation with using these chemicals. But it doesn't directly kill the bees. So it's not high enough to just kill these bees directly.
Have you seen any research where traps with borax and Crisco is used to control small hive beetle? Also, are there any methods used in Africa and Europe that are effective? Thank you for a very good presentation and excellent Q&A.
So the first part of that question, I'm not familiar with the research on using Crisco or borax inside the traps. For the second part of the question, was that in regards to control methods? Could you please repeat that?
Control methods are there any methods used in Africa and Europe that are effective as a control against small hive beetles?
So are there any effective methods using Africa and Europe?
Yeah, that are not being used in the United States.
Oh,
or not
okay.
legal to be used in the United States.
Oh, yes. Okay. So hmm, there are chemicals that a lot of beekeepers do use that aren't registered and aren't technically legal to use. Now, I'm not too familiar with the back end of this. Um, I could look into that definitely. I can drop my email in the chat. It's also on actually on this slide. If you'd like to shoot me an email, I can get back to you with some research on that, but I'm not too familiar with that.
This is an interesting question. What effect does a hurricane have on small hive beetles in hives or in general?
Yeah, so that's a great question. I actually saw this in, well, I don't remember the name of the storm, but it did hit in November. So we had a hurricane that came through. And so when we went back into the hives one week, and so this week of capture was completely disregarded. But when we went back into the hives, these traps were completely flooded. And so they had water up to the brim. And then the following week as well, when I went in to try and look for these beetles, the captures were a lot lower than usual. So it indicated that either it killed these beetles or caused them to become very, very inactive, but it did have some sort of effect within the hive. And the bees in general are also foraging much less. And the beekeeper I'm working with, he also noted that the honey that he had at the end of the year, he had about half the amount in barrels that he normally had each season. And so he thought the hurricane had a major effect on that because it did decrease the foraging activity during the honey flow.
Totally makes sense. Is there a treatment available or in development that would kill both small hive beetles and varroa mites at the same time?
So Comaphos is actually a treatment that exists that can do both of those things. So it's actually a dual treatment. So it does function to kill both varroa mites and small hive beetles.
Here's a question, comment sort of thing. If you ever want to go fishing with me and solve my beekeeping problems, hit me up.
I'd be happy to. Who is the person that said that?
I'll look
Where
it
they
up.
live?
Yeah. Maybe they can. I just let me see if I can pull up the question again. I just dismissed it. I'll look it up. I'll bring it
Sure.
up again. I
Uh-huh.
want to see if I missed any other questions. Should I be mixing beer with mineral oil?
So I didn't experiment with that, but one thing that I have noticed is that beer in general is a compound that evaporates very easily. So sometimes within 2 or 3 weeks, all of the beer can be gone. And so this is in Florida particularly, especially during the early fall or the summer. Now in cooler climates and going more into the fall, it might last longer, but you definitely could add either a quarter of the amount of oil or half. So you could do half oil, half beer, or a quarter oil and 3/4 beer. And so what this could do is serve as a buffer and probably limit the amount of evaporation that you're seeing. So it would cause the beer to last longer. I haven't experimented with that, but I could definitely see that working. And so a similar concept was used in the oil blend that was designed. And so the oil blend had an oil base and then also the volatiles from the beer. And so this causes that blend to last much longer than just beer.
Okay. All right. I think that's all the questions that I've seen. I'll come back to them and check again in a minute. But in the meantime, so as most of you know, Atreya was supposed to present last month, but we had to postpone it till January, which is sort of a good thing came out of it because in that last month, I've learned that Atreya has published a book. So I'm going to share for the book title and where you can get it. And we're actually going to have a raffle in a minute for a signed 2 copies of the book. So let me share that screen. I gotta find it. And that's right. As I'm sharing it, if you want to talk about it more, here we go.
Sure, yeah. So this is an independent project that I worked on. So I designed this and I wrote this completely independently. And so this is designed for children. So it's a storybook. It's filled with a bunch of different graphics, and it's basically an introduction to pollinators and bees in particular, how they benefit the environment and that sort of thing. So this would be great if you have any kids or grandkids, or even if you want to take a look. I greatly appreciate that.
So that's the name of the book. All right. So like I said, we're gonna have a raffle for 2 of them, and I put let me just make sure that I got everybody's names. So I put everybody's name who's in attendance into this little wheel of names, and we're gonna randomly pick 2 winners.
Can we make it 3?
3? Yeah, sure. We can make it 3. It's up to you. You're in charge here. All right, let me share that. Here we go. So if you're the winner, if you can send me an email, my email, I'll drop it in the chat too. Actually, I'll do it now. But my email is beekeeper@garfieldpark.org. And if you happen to be the winner, just send me an email with your name, address, and where you want me to mail the book to, and I will do that when I get it from Atreya. So my email is in the chat now. All right, so let's pick a winner. Here we go. Good luck, everybody. All right, Lawrence Williams, if you're still in attendance, please send me an email, and we'll get a book out to you. All right, winner number 2. Susan, same thing. Send me an email with your name, address, and we'll get it out to you as soon as possible. Oops, I forgot to remove Susan. If it ends on you again, Susan, I'll have to go to another person. Catherine Vickers, same deal. Please send me an email, and we'll get you the book, signed by Atreya. All right, let me stop sharing. Let me see if there's any more. Oh, one other thing that people have asked Atreya is, so what's next? Well, 2 things. First, is there a how can people follow you? Do you have social media account that you want people to follow or not? And then what's next for you? Are you looking to be more involved into honeybee research, pollinator research, or are you thinking about switching into something different?
So in regards to the first question, I'm not very active on social media, but I do have a LinkedIn, and I also have a YouTube. So I can definitely share those with you.
Yeah, please do if you want.
Yeah, sure. So I can share those with you in an email afterwards, if that's all right.
Okay, yeah, that's fine.
Sure.
I'll send it out afterwards. Sure. And then what's like what for your future? Are you thinking about doing more research on honeybees, or?
No, I definitely want to continue this research. So research in general is something I've just fallen in love with over the past few years. So I began my first science fair project in 3rd grade. So it was on strawberries. And so this was a very, very basic project, just looking at which temperature strawberries last the longest in. And since then, I've been continuing each year and trying to build upon the previous year's research. And so it's really just something that I love doing, participating in science fairs, conducting experiments, doing a data analysis, and writing up my findings and presenting my work is something I really enjoy. So I'm definitely going to be continuing this, and 10, 15 years from now on, I could see myself involved in research actively. So.
Excellent. We look forward to it. I mean, you're very passionate about what you do. So, as somebody mentioned in the chat a little bit earlier, I don't know if you happen to see that, but let me just read that. Your information, the information provided was very interesting, and I appreciate you taking the time to break some of the more scientific information into understandable, practical explanations. And there's a lot of that where the science part gets lost on beekeepers, because quite a few of us don't have the scientific background to understand it. So we look at the science and numbers, but don't understand the implications, and breaking it down, making it understandable is makes the presentation more memorable, and we absorb way more information that way. All right. I don't see any more questions. So before we leave, I just want to plug in that our next virtual beekeeping lecture will be with, let me share the screen, and if I have that pulled up, I do. So our next beekeeping lecture will be with Dr. Katie Lee from University of Minnesota on Wednesday, February 8th. And some of you may remember that she presented towards the end of 2021 on her research into healthy queens, healthy colonies, which was year 1 of her research. 2022 was year 2 and the final year. So she's going to present the updated data. It's not going to be a full data. She's not going to have all of the information available until about April, but she's going to have quite a lot more information than what she presented, and definitely 2022 included into it. So looking forward to it. It's the registration is open on our website, so feel free to sign up for it and join us on Wednesday, February 8th at 6:30 p.m. Central Time. So with that, Atreya, thank you very much for your time and for your presentation, and for signing the books and giving them away for a raffle. Very much appreciated. And for those of you who will read the book or buy the book, even for yourself or for grandkids, kids, if you could, after reading it, if you could leave a review on Amazon, Atreya would very much appreciate that. And let her know what you thought about it, and maybe giving some pointers on what his next book should be. So with that, once again, Atreya, thank you very much. Thank you everybody for attending. Happy New Year, and we'll see you next month.
Thank you so much. I really enjoyed it.
Goodbye, everybody.
Good night.